Hello everyone, my colleagues Vanessa and Philippe and myself are very pleased to present you today the PIM3 kinase inhibitor hit finding program. Let's put first the hit finding back into the drug discovery journey. The goal of the drug discovery process is clear. It's to identify the most promising hits and leads with drug-like properties in the shortest period of time. Successful clinical candidates are ultimately selected by employing the most robust and reliable hit finding strategies. The drug discovery journey is well structured with a well-defined sequence of steps: target validation, hit finding, hit to lead, and lead optimization. And all of these phases can be supported by our Eurofins discovery services. Let's now focus on the hit finding stage. The goal of the hit finding is to establish multiple tractable hit series for hit to lead phases. And we can approach it using four main approaches: knowledge-based, using the data of the literature; by virtual screening and computational science; fragment-based screening; and conventional high throughput screening, or HTS. These complementary methods are often done individually or in parallel and require a robust screening cascade in order to get high quality and tractable hits. Whether it's a new approach or a well-validated or an entirely novel target, HTS is a proven method to identify novel structural chemotypes. In this webinar, you will face a part of the team that will take care of your projects. I'm Céline Legros and I'm Partnership Director. I joined Eurofins Discovery 3 years ago and I'm highly involved in the design of the screening cascades for hit finding, hit to lead, and lead up programs. Previously, I worked as a scientific project leader in the screening department of Institut de Recherche Servier, designing and running screening cascade for HTS, hit to lead, and lead up programs.
Thank you, Céline. I am pleased to be here with you and Vanessa today. I am Philippe Dupuis. I have been working in the field of cell biology and in vitro pharmacology at Eurofins Discovery over the past 20 years. I was involved in many HTS campaigns, hit to lead, lead optimization, and safety projects. I'm here today in my quality of HTS expert to introduce HTS for hit triage and orthogonal assays for hit profiling. That's it for me. Vanessa.
Hi everyone, my name is Vanessa Porcolade. I'm head of Biophysics at Eurofins Discovery Cellular. I have a background in biophysics, working from fragment to antibody characterization. For several years now, I'm driving biophysics projects with my team in integrated drug discovery programs.
In this webinar, we will first go through the PIM3 project overview. Then we will take you through the HTS results, then the hit confirmation by biophysics assays, and finally the final characterization by orthogonal assays of the hits before concluding this webinar. So let's start with the PIM3 project overview. PIM are kinases firstly described as proviral integration site for the murine leukemia virus. The PIMs are a subfamily of three serine-threonine kinases, PIM1, PIM2, and PIM3. They have a conserved hinge region which create a unique binding pocket for ATP. The three PIMs show a high level of homology, 71% between PIM1 and PIM3 and 44% homology between PIM2 and PIM3. They are described as critical effector of the ABL, JAK2, and FLT3 oncogenes. PIM3 is reported as overexpressed in hematological solid tumor and adenocarcinomas. This makes of PIM3 a very attractive pharmacological target in cancer and immunotherapies. At the beginning of the webinar, I introduced you to the several ways hit finding can be addressed. With PIM3, actually, we have developed the following three approaches: conventional HTS, virtual screening, and fragment-based approach. Today, we will focus on the HTS approach. This project is supported by a screening cascade that combines multiple assays and technology: a primary activity-based assay, a biophysics confirmation assays, and then cellular target engagement. What is less conventional is to include early selectivity panels, Here, PIM3 versus PIM1 and PIM2, and early ANME assays that will help in the selection of and prioritization of the hits for future programs. Now, Philippe and Vanessa will present you the scientific data of the PIM3 program.
Thank you, Céline. So I will say a few words about HTS used for hit triage for compound activity. So, as Céline introduced, in this project, our goal was to find active hits for PIM3. We also wanted these hits to be selective for PIM3 over two closely related kinases, which are PIM1 and PIM2. For this purpose, we screened two urethanes compound libraries, one diversity library of 50,000 compounds and one kinase-focused library of 23,000 compounds. So the expectation, the specification, and in other words, the target product profile of our hits were the following. They had to be active hits in a primary assay with a PIC50 above 5. They had to be confirmed in orthogonal activity assay. They had to be also confirmed in orthogonal binding assay. The opt-in hits must have tractable chemical properties. They have to show activity in cellular contact and a reasonable selectivity for PIM3 versus PIM1 and PIM2. And last but not the least, we wanted them to have a good ANME properties. I will now say a few words about the HTS platform and approach. The recombinant active PIM3 kinase enzyme was produced in-house, which is a great advantage. We use eco nano dispensing in 384-well format to prepare the ready-to-use plates. We develop in-house the primary assay using the adaptable technology from Pomega and use a Ferrastar reader. As you know, Eurofins has a great expertise to develop assays. We did it thousands of times over the 30 last years. Eurofins' strength is also its ability to miniaturize and industrialize assays to make them high-throughput compatible. We indeed adapt the level of automation based on the need of each project. We can use either semi-automated process combining workstations or fully automated platform. This ensures efficiency and robustness. I will now introduce what we executed at the bench and share with you the key figures in terms of results and data analysis. So we first start, of course, by a pilot screen to challenge the robustness of the assay and also to predict the hit rate of active compounds. For this purpose, the pilot screen was performed using 20 plates randomly selected from Eurofins' Discovery Diversity Library. This represents a bit more than 7,000 compounds. They were tested in syndicate at one concentration of 10 micromolar phenol in two independent experiments. As you can see, the signal windows was pretty comfortable with the SNR around 40 and the Z prime factor over 0.8. The SNR and Z prime were stable and over the standards. When we look to the correlation between the two independent experiments, we can see that there is a perfect correlation between the percent inhibition obtained in first and second experiments. The assay is repeatable and robust. On the graph at the bottom of the slide, we reported the distribution of the activity of the test compounds. Each bar represents the number of compounds which display the given percent of inhibition, from the less active on the left to the more active on the right. We observe a Gaussian-like distribution. We can then calculate the mean activity value. We can also calculate the standard deviation. And when we apply the mean plus 3 standard deviation, then we define the selection threshold to apply. Any compound above this threshold can be considered to be active compound. With this low selection threshold, the hit rate is estimated to be close to 1.32. So the pilot screen results gave us a clear go for the one-dose screen of our 71,000 compound library. They were tested in syndicate at one concentration of 10 micromolar phenol in a single experiment. As expected, the signal windows was still comfortable with the SNR around 35 and the Z prime factor still over 0.8. So the assay performance perfectly aligned with the pilot. When we look at the distribution of the activity of the test compounds, we still observe a Gaussian-like distribution. and we calculated a selection threshold of 31%. So any compound which display more than 31% inhibition can then be considered as an active compound. Based on this, we selected 1,000 active compounds, which correspond to a hit rate of 1.54%. The hit rate is a little bit higher compared to the one obtained during the pilot screen, due to the fact that the diversity library includes a kinase focus set. As a conclusion, we can say that the one-dose screen was as good as the pilot screen. The results were perfectly aligned with the pilot screen data, and 1,000 compounds were selected for dose response curve. So let's have a look to the dose response curve results and analysis. In order to determine IC50 and PIC50, we ran dose response curves. The 1,000 compounds were tested in duplicate at 8 concentration in 2 independent experiments. The signal windows was maintained with a SNR around 27 and a Z-prime factor over 0.8. We get the same robustness as for the previous steps. When we look at the correlation between the 2 independent experiments, we see a perfect correlation. All this means that we can trust the hit we selected. If we report the compound PIC50 frequency of distribution, we can say that we determine IC50 value for 946 compounds over the 1,000 selected and tested, and we get PIC50 from 3.4 to above 7.5. So it means that the most potent compounds are in the 10 nanomolar range. So it was pretty successful with 94.6% confirmation of the effect. Then, with the help of our medicinal chemists, based on the potencies and the scaffolds, 140 compounds were selected for secondary hit triage assay. We were really happy with this. So as it is a teamwork, I will now let Vanessa to speak about biophysics and hit triage.
Thank you, Philippe. Now I will show you how biophysics could be used as second hit triage in the PIM3 project. As an introduction, several technologies can be used as a triage for compound binding. Here at Eurofins Discovery, we are working with the most recent technologies. From the left to the right, you can find the BIACOR 8K+, the Dianthus, the PIC ITC, and also the BioRad devices. All of those technologies are complementary and have some advantages and limitations. In our PIM case study, we are performing a feasibility study using SPR and MST spectral shift. Before going into details, I would like to highlight that all of those technologies are using specific setup and readout for binding characterization. However, they share common specificities that are the need of highly pure and active recombinant proteins. They will be used for immobilization, for example, by SPR, or they will be labeled with a fluorophore for MST, or used as well in solution for ITC. In our PIM3 case study, specific recombinant protein constructs were designed by our colleagues at Eurofins Discovery. We have to keep in mind that the orientation of the tag on the protein can be crucial for binding studies. Moreover, the quality of recombinant protein should be higher than 90% to increase our chance of success. After the HTS dose response confirmation that was just explained by Philippe, the biophysics cascade is usually divided in 4 steps. With a feasibility study, an assay development with a reference compound, a single dose experiment, and then finally the dose response curves. Now let's have a look into more detail about the feasibility study using SPR and MST spectral shift with PIM3. The first technology used for feasibility study is SPR, standing for Surface Placement Resonance. It's a gold standard for studying the molecular interaction between 2 partners in real time. In our case, the protein is immobilized on the sensor chip and the compound is injected in the microfluidic system. If a binding event occurs, a response in resonance unit will appear with an association phase, an equilibrium, and then a dissociation phase, as depicted on this picture. From this sensorgram, we can extract by steady-state affinity model a KD value, or using the 1-1 binding model, we can extract kinetic parameters K on and K off. As additional information, with our BioCorrect K+ having 8 channels, we can test up to 1,000 compounds per day in single dose and up to 50 compounds in dose response. So now let's have a look on our Dianthus for MST spectral shift data. The second technology we use here in our PIM3 case study is a microfluidic free screening platform. In that case, the protein is in solution and labeled with a fluorophore. If a binding event occurs between PIM3 and the compound, a modification of the fluorophore environment will occur due to a ligand proximity or a protein conformation change. 2 complementary readouts could be used at this stage as well. TRIC for temperature-related intensity change or spectral shift. The TRIC measures the strength of the interaction by detecting a variation of the fluorescence in function of the temperature change. From a MST trace with a significant amplitude, we can extract a KD value. Alternatively, the spectral shift detects after an excitation at 590 nanometers a shift of spectrum toward either blue or red. The detection of spectral shift is achieved by a calculation of the ratio between 670 nanometer over 650 nanometer. We can then extract a KD value. Regarding the throughput of this technology, the Dianthus offers a higher flexibility with lower sample consumption compared to the BioCorrect K+. We can test up to 1,000 compounds per day in single dose or up to 150 compounds in dose response. So now, having introduced you the equipment, I will show you the experimental data for PIM3 feasibility study using both technologies. Before starting screening compounds, we need to perform a feasibility studies to check how the protein is behaving in each technology. You will find on the upper panel the SPR data and on the lower panel the MST spectral shift data. The non-hydrolyzable version of ATP is used as reference compound for PIM3. Here, the aim of the feasibility study is to compare the setup for binding studies. PIM3 is immobilized on the chip for SPR, whereas MST spectral shift, we have PIM3 in solution coupled with a non-covalent fluorophore. Regarding SPR data, our internal control behaved properly. However, no binding was observed for PIM3 and ATP gamma S immobilized when PIM3 is immobilized in an orientated manner or in a random way with amine coupling. Interestingly, we start to see a negative dose response that suggests that ATP is also binding the reference surface. And as you can see on the right, no binding is observed on the reference surface. The outcome of this SPR feasibility study show that the signal variability may be induced by a PIM3 structural change upon binding with ATP. This phenomenon is quite often observed for other classes of biomolecules like aptamers. Let's focus now on the MST spectral shift data. The fluorescence of PIM3 labeling is monitored with the initial fluorescence parameter. As you can see, it's stable over the different concentration of ATP. For spectral shift data, a nice dose response was obtained in duplicate and small standard deviations are observed, meaning that the spectral shift gave us robust data. It's not the case for MST trace, where we can start to see large standard deviations. An optimization of MST buffer conditions need to be performed for this assay. Based on those preliminary data, we selected the spectral shift for the second hit triage in the PIM3 cascade. For the second step, the assay development will help us to select the right labeling strategies, to optimize the screening buffer, to ensure the protein integrity and the data robustness. The his tag for PIM3 was labeled with a non-covalent dye using 2 labeling buffers, including the protein storage buffer. The titration of PIM3 with the dye showed KD lower than 10 nanomolar for both conditions. However, aggregation started to be observed with some concentration using the protein storage buffer. That's a typical example where it's relevant to perform a buffer screening. The protein buffer storage is not always a buffer of choice for binding studies. Then regarding screening buffer, the assay development turnaround time with spectral shift is reduced. In fact, the buffer conditions don't need a lot of optimization like MST. This assay development was performed in less than 2 weeks with an excellent data robustness using ATP gamma S as a compound reference. On the graph, you can see all of the KD value of ATP gamma S for PIM3 over the different assays. So now we are ready to test the 140 compounds coming from the ADP-GLOW assays. So here we moved directly to the latest stage of the biophysics cascade as we are working with less than 200 compounds. For those response curves, the compounds were dispensed in nanoliter using an echo in specific nanotemper 394 well plates. After validation of the assay with ATP gamma S, 92% of the compounds for PIM3 kinase. Interestingly, 22 compounds showed already a nanomolar affinity. It will be now interesting to compare the binding data with the previous ATP-GLOW activities with those compounds. Here on this graph, you will find on the x-axis the mean PKD value from spectral shift data, and on the y-axis the PIC50 from the ADP-GLOW assays. As you can see, no correlation is observed, and that is expected as there is no linear correlation between an IC50 and a KD value. Biophysics allows us a finer ranking of the compound that have a similar PIC50. As you can see in the plot, we have a range of 3 log units in spectral shift PKD for 1 log unit in the PIC50. In our PIM3 case study, the binding constant generated by biophysics was used as supplementary criteria by medicinal chemists to analyze and select the best series for further profiling. I now hand over to my colleague Philippe, who will describe the orthogonal assays for PIM3 hit profiling.
Yes, thank you, Vanessa. So after hit triage, I will now say a few words about orthogonal assays for hit profiling. Orthogonal assays for hit profiling were indeed conducted using both mass spectrometry with rapid-fire injection for activity assay and NanoBRET for cellular target engagement. Today, I will focus on NanoBRET and confirmation of compound activity in cellular context. So here again, we fully developed the model. The 140 compounds were tested in duplicate 8 concentrations in 2 independent experiments. The signal window was fine, and also the Z prime factor pretty good at 0.83. If we look at the graph on the left, we can see that the correlation between the 2 independent experiments is very good. The assay is robust. If we look at the graph on the right side, we can see that the correlation between the ADP-GLOW and the NanoBRET result is good also. With the NanoBRET, we are in a whole cell model. and so, not surprisingly, we observed a shift of 1.24 if we consider the PIC50 values, which means that compounds are 1 log to 1 log and a half less potent in NanoBRET than in adaptable. As a conclusion for this orthogonal confirmation step, about 50% of the hits were confirmed, which give us 67 compounds showing activity in cellular context. One more time, with the help of our medicinal chemists, 16 hits over the 140 hits distributed in 6 clusters has been selected for the following steps. If we summarize the HTS campaign and look at this inverted pyramid, we can say that we developed several models based on ADP-Glow, NanoBRET, mass spec, and several biophysics assays. We went through pilot screen, one-dose screen, dose response curves. We did the orthogonal screen in biophysics and also in cellular context. Thanks to this hit triage and medchem analysis, we successfully identified and characterized 16 hits distributed into 6 clusters from our 71,000 starting compound. We went further in this program. In addition to the pharmacological properties, our hit finding programs took advantage of the Global Eurofins Discovery Service offer. Our medicinal chemists indicated that the hits are chemically tractable based on their scaffolds and their chemical properties. In terms of chemical properties, we speak about their molecular weight, the calculated partition coefficient, the hydrogen bond donor, the hydrogen bond acceptor, and the presence of heavy atom. Concerning the selectivity assays, we use the Eurofins Discovery's Kinase Profiler and KinomScan activity platforms to investigate the selectivity of the 16 hits against PIM1, PIM2, and PIM3 kinases. Kinase Profiler is an activity-based platform, and KinomScan is a binding-based platform. All the 16 hits were confirmed, and 2 scaffolds show selectivity for PIM3 versus PIM1 and PIM2. Moving to ANME, we know that it is of a great help for series prioritization for hit-to-lead program. We indeed took advantage of the Eurofins Discovery ADME platform and ran a small ADME package in order to evaluate the compound aqueous solubility in PBS, to assess the lipophilicity expressed as the log-D partition coefficient, and also to evaluate the mouse and human hepatocyte stability. This gave us the estimation of the interesting clearance of the compounds and their half-life. All this work allows us to successfully identify 2 chemical series as priority 1 for the future hit-to-lead programs. I stop here with this very promising review and I pass it back to Céline.
Thank you very much, Vanessa and Philippe. My colleagues took you through the PIM3 screening cascade that combines multiple assays. Assays we can consider as a toolbox to support HTS program. I mean protein production, robust and high throughput primary assay, biophysics assays are SPR or MST TRIC spectral shift, orthogonal activity assays that haven't been presented today, cellular target engagement, selectivity platform with the kinase profiler and the KinomScan, and then off-the-shelf ANME assays. It's really important to have access to a wide variety of assays to get high-value hit series. So the whole toolbox is really there to support HTSs, but not just conventional HTS, as it's described on the screening cascade. Assays, having all those assays in place for HTS would also allow to combine the other hit finding approaches. As the fragment-based screening, fragment that will be first screened in biophysics assays, but could also be included into the small molecule library in the primary activity assay. Similarly, compounds designed by virtual screening could be directly plugged at the stage of the single dose or dose response curve of the primary screening assay, depending on the number of compounds to be tested. We have now a few take-home messages coming from this webinar. An hit finding can be addressed based on several approaches that are complementary and could rescue a project for complex targets. It's important to draw a comprehensive screening cascade, including orthogonal assays, especially based on different technology. For our customer, we design screening cascades specific of each project and target. We try to keep innovation by integrating the most advanced technologies, are the MST-TRIC spectral shift today, or also the EcoMS, high throughput mass spectrometry screening technology. And this to accelerate the drug discovery as much as possible. If you would like to learn more about the HIT series of the PIM3 programs or about our HIT finding and integrated services, please contact us at email address hitfinding@eurofins.com.